Hi, my name is Jakob, and I want to show you how to proofread the H01 dataset using Cave. So first, you'll have to go to the H01 website, and under the tab called Proofreading, you'll find a detailed written tutorial with instructions and everything that I'll also show in this video. But before you can start proofreading, you have to click on this form and answer a bunch of questions, and we'll usually get back to you within 24 hours, and then you are all set to start proofreading. To get started, you can just go to ngl.brainwire.org, and that brings you to an empty Euroglancer window. You first have to log in by clicking on the login button at the bottom of the page, and that assumes that you have signed up and been approved for an account already. So to start proofreading, you have to click on the top right on this volume button and select the H01 dataset and click confirm afterwards. And then you'll see the imaging data plus the automatically generated segmentation. And now let's see if we can find a neuron where I can demonstrate proofreading to you. So I'll just zoom in, and I think maybe this one is a good candidate to start proofreading. So I'll click on it, and by zooming in the 3D view, I'll see the three-dimensional structure of the segment. And my approach usually is that I take a look at the ends of the segment, and here I can see that there's probably something cut off. So yeah, here, this is a good example of a split error. So those two segments were incorrectly split, even though they belong to the same neuron. So I'll select the other segment that belongs to that neuron. By clicking on it, I'll have a look in 3D whether they actually seem reasonable to belong to the same segment. And hitting the L key, I can recolor the segments to show that they're now actually they're two different segments. And by clicking the M key, I can enable the merge menu. Okay, and now you'll see that you get an error message when control clicking on the segment. And to fix that, you can just right click on the segmentation tab on the top. So you have to have the segmentation tab enabled in order to be able to proofread. So when you now do control click on one neuron and control click on the other neuron, you can connect them using this red line. And that means that you have scheduled a merge to happen between those two segments. But before we actually execute that merge, let's see if we can find another split error. So I'll jump to the other end of that neuron and see if I can find a continuation. Yeah, that's a good view. There it is. I selected it in 3D and connecting it again using control click on both ends of the segments. Let's see if we can find another one. All right, there it is. Yes. And now again, doing control click on both ends on the segments, I can connect them. So now we have scheduled 3 merge operations to happen, and we can execute them either by clicking the submit button in the merge menu or just by clicking enter. Now you see the colors of the segments change and also the IDs change. And if you give it a second, you'll see that now everything was merged together, and also the meshes between the merged segments are now recomputed. So that's how you perform merge operations using Cave for the H01 dataset. Now I want to briefly show you the proofreading changelog. So if you click on the segment tab, and then you'll see these 3 dots next to the selected segment. And when you click on those, you can see a link to the proofreading changelog. So that will open a new tab, you can see that for that particular neuron that we just selected, 3 proofreading edits have been made. All of them were merges, and they were performed by me. So the reason why this tool exists is to just make sure that everybody who invests a lot of time in proofreading can get credited for it. So for each neuron, everybody can always look up who worked on it before. All right, that's it on how to fix split errors by merging segments together. I will share another video as well on how to cut segments apart to fix merge errors. See you soon.